I'm representing here the Center for Biologics and Evaluation and Research, and specifically the group that regulates gene therapies for a variety of conditions, including hemophilia A and B. So my disclaimer, I don't have any conflicts of interest, but I will not be able to discuss any specific products under investigation that are being reviewed by FDA. I'll talk in general about all products. So the outline of this presentation, I'm going to spend a little more time discussing about the primary endpoint, selection of a primary endpoint in clinical trials for registration, for marketing application, and the challenges associated with this endpoint selection. And then talk a little bit about trial design considerations, also as related to the selection of the trial endpoint. And then finish with a few comments about long-term safety monitoring and long-term efficacy monitoring. So the FDA has a number, as does the EMA and other regulatory bodies, a number of programs to expedite promising products. We currently have for all products, drugs and biologics, four expedited programs: fast track designation, a breakthrough designation, priority review, and accelerated approval. And for gene therapies, we have a fifth, and cell-based therapies, a fifth expedited program called Regenerative Medicine Advanced Therapies Designation. And, but I'm going to focus here more on one of the programs, which is the accelerated approval, and explain a little bit what this means in regulatory terms and what this means for access to promising products. So for any of those programs, the two criteria that are common to all of those is that the condition be a serious condition and that there will be an unmet medical need. And serious condition means it's a condition that is either fatal or affects substantially the day-to-day functioning, or if the condition is not treated, that it will lead to a more serious condition or death. By this criteria, hemophilia, all forms of hemophilia, particularly severe hemophilia, meets that criterion. Unmet medical need, we have to realize for hemophilia that there are some safe and effective therapies, both clotting factor concentrates, the extended half-life now available, and the non-factor therapies are already available in the US and in developed countries. There's a substantial unmet medical need in other countries, in the rest of the world. And in this World Federation of Hemophilia Congress, it's something that we need to acknowledge and recognize. But we are talking about unmet medical need in the United States. And so still we can identify that there is some unmet medical need in certain groups, in severe hemophilia, with still existence of bleeding, substantial bleeding, and teenager compliance and other groups compliance with this frequent therapy, whether it's 3 times a week or every 4 days or twice a week or once every 2 weeks. It's still a complicated regimen for a lot of people to maintain, and to maintain only levels of factor 8 or factor 9 that are just above the severe level. So there is an unmet medical need here. And the expedited program that I'm going to focus on is the accelerated approval. What this means is if you have a traditional approval, it's based on a clinical benefit endpoint. So the endpoint is directly translated into a clinical benefit. Clinical benefit, what I mean is how patients live, function, or survive, and so it's a direct measurement of how they function and or whether they survive or not. That's a very straightforward endpoint. And once a drug or biologic undergoes traditional approval, this is done. There is post-marketing commitments, post-marketing studies that are needed, but there's no further confirmation of that benefit necessary. With accelerated approval, it's typically based on a surrogate endpoint And it's a kind of a conditional approval. In EMA, the equivalent would be a conditional approval. It's a tentative approval based on a biomarker. And if that's the case, if that's a pathway for approval, that usually requires confirmation, a clinical study to confirm that there is a clinical benefit associated with this. Another way to have accelerated approval is through an intermediate clinical endpoint. So that's a measure of direct clinical benefit, but it's measured at a point earlier than what we would like to see for a durable and significant effect. So let's talk first about surrogate endpoint for hemophilia. The obvious surrogate marker or biomarker here would be the factor activity level. As we discussed all through the meeting, one of the pros of having an accelerated approval based on this surrogate endpoint is that it's a direct measurement of the factor being replaced, whether it's factor 8 or factor 9. It's more objective. It's a number. You can say it's the mean is 15% or 30% or 50%. And it's also something that we use to manage clinically patients. So when we titrate factor replacement, we titrate to a specific level or based on pharmacokinetics to achieve a level that will not cause bleeding. The cons is that we have a lot of experience now with on-demand treatment and perioperative management with factor concentrates and even with extended half-lives in terms of prophylactic treatment. But those have peaks and troughs, and we're not sure what would be an adequate steady-state level. You see that even with the gene therapies, there's a usual bump in factor activity, and that drops and that remains somewhat stable over time. We don't know what is the right factor level that is adequate to decrease or prevent further bleeding or to correct the problem. There is also interpatient variability. So one patient may be doing very well with just above a 5% threshold, and others will continue to bleed with a 20%, particularly if they have severe hemophilia with target joints. So it's unclear how this variability between patients will affect the selection of a particular target threshold for an approval based on that factor activity level. Also, Dr. Peace alluded to the issue with the right assay. What's a chromogenic assay is discrepant usually from the one-stage clotting assay, and even between labs there are some discrepancies, and so it's unclear to us what if this is a threshold based on a factor level, what would be the right assay. Finally, typically surrogate endpoint is something that we would consider to expedite approval because it's something that's measured earlier than a clinical benefit. An example was the initial accelerated approval. The first accelerated approval that we had for HIV treatment for AZT was based on a laboratory measure, not on survival for HIV infection or AIDS. This because CD4 counts and HIV counts could be determined much earlier than mortality in AIDS. But here, when you talk about annual bleeding rates and factor levels, they are determined at the same time. And so it's unclear what would be the advantage. Some people would say, well, but there is more precision in factor levels, so the power of the study, statistical power is higher, so it will lead to being less patients to have the same kind of precision. But that's an argument that we can make. On the other hand, the clinical benefit endpoint is mean annualized bleeding rate or bleed rate. And why is this important? Because that's what is the cause of the morbidity, and in early days in the US and still in a lot of the world, a cause of mortality of patients. So, and that translates into a direct benefit. So less bleeding or no bleeding as we've seen more recently, at least as a mean, is a significant benefit. There's an advantage also that's patient-reported, so there's no need to rely on laboratory measures. It's something that we can get from patients in the report. We have available historical data, so we know the history in terms of factor replacement, what the bleeding rate is, and particularly bleeding rates for that particular patient. The cons of the annual ABR or annualized bleeding rate is that exactly one of the benefits is that it's patient-reported, but one of the disadvantages is that it relies on patient reporting too. So there is some overcalling, there is some, we've seen even in this meeting that there is some discrepancy between patients, particularly with target joints, that are calling a bleed when it's actually an exacerbation of pain for other reasons other than a recent bleed. And also there is an issue with compliance. If those trials last for a year or 2 years and patients are required to keep complete records of their bleeding rates, then there's an issue with how adequate that compliance and dependence on patient reporting would be. It's also somewhat subjective in nature, as I alluded to. What happens when you start with a mean ABR that's 0? How much better can this go? So that's another issue with this particular clinical endpoint. Let's move to trial design considerations now. So again, this all pointed towards the design of the study based on the selection of an endpoint, be it surrogate endpoint or a clinical endpoint. For eligibility, you've seen here, this usually for adults, not for children, at least for the AAV therapies that we've seen, the vector therapies, because you require a certain level of liver maturation and lack of hepatocyte division for this to work. It has to be in seronegative, AAV seronegative patients, because we don't expect that AAV seropositive patients will have any chance that the gene therapy will work, and they have to have normal liver function and no impairment based on prior history of hepatitis C virus infections, for example. But focusing on the levels of activity, factor activity, you have to have a very well-established level. So patients have to have severe hemophilia, meaning before treatment their factor levels were less than 1%, and they have to have a history of bleeding prior to factor replacement therapy, and they have to have well-documented factor therapy, whatever the form of the factor replacement therapy is. There is also the issue that I alluded to before with interpatient variability. So if you select a threshold that's too high, there's a risk that the trial will not meet that threshold. Let's say if you say, well, I want to select a threshold of 30%, that's going to be considered a responder in my clinical trial. There is a chance that there will be not enough responders for the trial to be positive for regulatory consideration, but that doesn't mean that 30% is critical for a patient that may stop completely any bleeding with far less factor replacement on a steady state level. So that's another consideration when designing a trial in terms of the sample size and adequate statistical power. With regard to an annualized bleed rate, you have to have a patient that has well-documented factor replacement therapy for a lead-in period, be it 6 months or a year, where the dose of the factor replacement, the clotting factor, has not been changed substantially, and particularly in the few months prior to the treatment with the gene therapy, for us to be able to interpret any results. And also documentation of the annualized bleeding rate prior to gene therapy. So there will be an adequate comparison between before and after gene therapy of that annualized bleeding rate. there is a question about what's the adequate non-inferiority margin. Typically, we say we expect the non-inferiority margin, the upper bound of the non-inferiority margin, to be not exceeding what the patient had before, or as a group, as a mean non-inferiority margin for ABR, and not exceeding the conventional clotting factor replacement or the extended half-life factor replacement. So we expect even that there are potential risks of gene therapy that this would be no worse in terms of ABRs than what's on the market. And then there's the issue of co-primary endpoint. As Dr. Pierce alluded to before, EMA has been more accepting of factor activity levels as a primary endpoint. I think the FDA is somewhat discussing this, but is moving towards that direction, although it's not anything that is conclusive at this point. But regardless of this, some people say, well, how about if we propose a co-primary endpoint of factor level as a surrogate plus ABR as a clinical endpoint? We think that that's going to be a burden on companies and sponsors to achieve. It takes more patients, and the trial will be longer because of that if we consider co-primary endpoints. So one solution would be do a trial with an early endpoint that is factor level replacement. Once, it's very important to say factor level replacement at 1 year after achieving steady state, because you see for the first 12 weeks or even longer sometimes, you have fluctuations of that factor level. But once there is some stability on the factor level, if the company can show us that there is durability of that factor level activity, that could be a consideration for an endpoint. And then ABR could follow, even if it's not a secondary endpoint, if it's a primary endpoint, in a hierarchical manner, ABR would be demonstrated. Now, with what the commissioner had said, it's possible that we would be moving this to a post-marketing commitment or requirement for demonstration of the clinical benefit. And let's move gears now with the time that we have with what we require for long-term safety monitoring. So it's not very clear yet, even though we have a number of clinical trials from different companies about the adverse event profile. The number of patients in each trial is relatively small, and the duration of follow-up is not quite robust for us to be able to determine the safety of each of these products. So, of course, we are worried about the development of inhibitors. If there's a new inhibitor, someone who has no inhibitors prior to gene therapy, we need to capture that information. And as well as any developments of malignancies, as Dr. Pierce and others here during the meeting have alluded to, there's a small risk of integration and oncogenic potential, other neurological changes with the high dose of AAV administered systemically that could happen in other autoimmune disorders. So we have guidance. The guidance for lentiviruses and retroviruses is the requirements usually for 15 years. But we believe that for AAV, given the smaller risk of integration, that safety monitoring would be 5 years would be sufficient. And that's what we've been telling sponsors. It doesn't mean that sponsors will only come in with a marketing application at 5 years of follow-up. They can come in much earlier, but the commitment to follow each patient in the study would be 4 or 5 years. Now, we want to adopt a least burdensome approach to data collection. We want patients to be retained for this long-term follow-up. And we understand that if you have very invasive, very frequent monitoring, there will be a loss of data that's precious data for us to understand the safety comprehensively. So at this point, we think that a visit to look at how patient feels and factor levels and inhibitors and liver enzymes, a check of liver enzymes is sufficient. We don't believe that liver biopsies are sufficient, are necessary. They're burdensome, and they could be affecting retention, and they could be risky. So in terms of long-term monitoring for efficacy, we're very interested in the durability of effect, how long this improvement in factor activity level will last, and if there is any drop in factor activity level to below 5% or 1%, what are the consequences? Can patients be retreated with extended half-life factors or clotting factor concentrates or other therapies and non-factor therapies? And how is this effect after having a gene therapy? We also need to know how gene therapy, after stable factor activity levels, how does it affect the management, clinical management of patients that experience trauma that can increase the risk of serious bleeding, or patients that have perioperative management for surgery, prior to surgery and during surgery, and how gene therapy will be affecting that. And more importantly, we want to look to the future as something positive and look for what happens to stable joints. Is the chronic pain going to go away gradually since there is no more bleeding? And what the future would look at for patients on a long-term basis. So with that, I thank everybody for your attention.